Okay, we are recording. Excited.
Hi, well, so nice to meet you all, and see, there's a lot of young faces here, so that's really exciting to see. I'm glad you're all interested, and thank you for having me at the working group. So I'll be presenting some of my work, not all of it, especially recently, but still works that I'm kind of extending and still thinking about now. Title it would be Novel Methods for Statistical Analysis of Covariance Structures, and this is a presentation that will span a couple different types of data and a couple different types of covariance structures, which I'll outline before I begin. So first of all, why do we care about covariance in the context of structural neuroimaging? So in structural neuroimaging, we tend to acquire entire brain images, generally through MRI, although there are many other possibilities now. But the idea being that we want to measure some kind of feature on distinct regions of the brain. And so here I just show an atlas just describing the right-left hemisphere, lateral medial views, and kind of highlighting some of those areas that could be defined. And what we may be interested in at specific regions could be something like the volume, the total size, some cortical structures, including sulcal depth and cortical thickness, for example. And all these kind of region-specific features can give us some information about the disease. So for example, in Alzheimer's, it's very well known that the hippocampus is impacted both in the volume and the cortical structure very early on. But what might be a little lesser known is that actually the degradation in Alzheimer's is somewhat asymmetric, where the left side of the hippocampus will degrade slightly faster and earlier than the right side. And when it comes to a kind of pattern like that, it becomes not as obvious how we kind of measure that kind of degradation. And so the way I like to think about it is that's actually a kind of change in the correlation between the right and left hemispheres. And in that case, so like for example, as one degrades faster than the other, they will no longer be as kind of tied to each other in a population. Correlation will dip. And so that's how I see covariance being important in structural neuroimaging when it comes to certain kinds of patterns like that occur across the brain and between regions of the brain. It's important to consider essentially the covariance or correlation of those brain regions in structural neuroimaging. And this can give us important information about disease and about various other kind of trends in the brain. But a covariance structure is important in other contexts as well. And so one of the other contexts, which a lot of you seem like you're very familiar with and very interested in, is in functional neuroimaging. So let's play this video to kind of that kind of highlights in a visually appealing way what I'm talking about here. But we use functional imaging, specifically functional magnetic resonance imaging, to kind of capture activation in the brain across time. And what that can tell us, and as you see in this video, is that certain areas of the brain will kind of co-activate at the same time. And this will happen across different tasks or even just across just at rest. And so certain brain networks can be derived and kind of described through the measurements that we acquire through functional imaging. And again, this comes down to a covariance structure, but this time within a single individual and between functional regions of the brain. And so for example, here you'll see that certain regions tend to be correlated more with each other across time. And this is in a resting state, so no task. And this kind of covariance structure can tell us a lot about the brain and about the brain organization in particular. And then finally, more generally, including both structural and functional neuroimaging, we care about covariance because it can describe the relationship between different types of data. So for example, if we wanted to see how structure and function are related in the brain, or beyond that, see how structure and function in the brain may be related to other kinds of data that we can collect. For example, transcriptomics or microbiome data, EHR, like wearables, all of these kind of new types of data that more and more are kind of being collected together in large scale in large scale studies. And so the question of interest is how these data types are related, and if we can leverage these data types together to tell us more about certain outcomes, things like personalizing healthcare or neurodegenerative disorders. Can these types of data altogether tell us something else? And the way we capture this can be through a covariance structure, but this time across different types of data. So for this talk, I'll be trying to highlight some work I've done in all three of these areas, but I imagine that to leave some time for discussion and just kind of focus in, I think I'll mostly be focusing on the structural and functional neuroimaging, and maybe only briefly touch upon the kind of project in the multiple data types. So in the context of structural neuroimaging, this is a project that kind of looks at statistical harmonization for reducing side effects in the covariance of multivariate observations. And this project is motivated by studies of Alzheimer's disease. Uh, so Alzheimer's disease as many of you may know, is a very prominent public health issue. This is because it's the seventh leading cause of death in the United States, and projections show that the number of cases of Alzheimer's disease is likely to more than double by 2060. And currently, it's about 6.5 million, and we're looking at something like almost 14 million cases projected by 2060. And the burden on the healthcare system is only expected to increase. And because of this need, a lot of research has been dedicated to better understanding Alzheimer's disease, the progression, and ways that we can perform early detection, for example, in order to administer treatments as early as possible. And so all this research has led to the discovery of a large array of biomarkers that occur at various time points in the course of the disease. So I'd just like to highlight the x-axis here to talk about some of these definitions. So first of all, we consider Alzheimer's disease to be pre-symptomatic prior to any memory impairment. And then as there's kind of early signs of memory loss measured using cognitive tests, we call this mild cognitive impairment, or MCI. And then finally, Alzheimer's progresses to kind of the well-known symptoms, which include even more severe memory loss, and also impairment to daily activities and loss of automatic functions. And that we call dementia. So there's a lot of different biomarkers, some of them collected, as, some of them involving biospecimen, and some of them less invasive that involve imaging. And so I'll be specifically talking about the MRI-based biomarkers for Alzheimer's disease. And there's a wide number of these. And I talked about a little bit about how the hippocampus can change across time, including brain volume, atrophy in specific areas, white matter hyperintensities, and another property is cortical thinning. And I'm actually going to focus on cortical thinning for this topic. So to define cortical thinning, I'm going to go through and explain how we measure cortical thickness. And that's just the thickness of the outer layer of gray matter in the brain. So typically, this has been done using automatic methods. So I'm highlighting a figure from an early paper that quantified cortical thickness, just showing that we use kind of quantitative measures to, first of all, segment the gray matter on the outer layer, and then approximate the thickness at various regions of the brain. And this thickness varies quite a bit depending on which region you're considering. But typically in a healthy brain, it will vary between 1 and 4.5 millimeters across the cortex. The variance is quite high. And in the context of Alzheimer's disease, studies have found that, again, using quantitative measures, that we're able to note a degradation of about 0.2 millimeters in certain areas of the brain, and this is in mild cognitive impairment. So this is important to look at because if we were to use structural MRI in order to detect Alzheimer's earlier, it'd be important to kind of detect these kind of changes, and specifically note in certain regions, in particular on the bottom, you can see this is kind of where the hippocampus and a lot of areas related to memory are. So it's really important to try to be able to detect this difference of 0.2 in a healthy brain where we're looking at a scale of up to like about 4.5 millimeters. And I'd like to present one more study, but I'm not going to give you all the details about this study. All I'll tell you for now is that this study consisted of 14 subjects scanned 1 month apart, so looking at longitudinal changes. And um, what I haven't told you yet, though, is that are these subjects Alzheimer's disease patients, or are they just cognitively normal? So based on the figure alone, looking at this rate of change, which is above the scale noted in mild cognitive impairment, and in certain regions that are notably impacted in Alzheimer's disease, it may seem like this is an Alzheimer's brain. But the surprise is that these subjects are actually all cognitively normal. And the last detail about the study that I didn't tell you is that these subjects were moved between 2 different MRI scanners across that 1 month period. And so just looking at that, we noticed differences essentially entirely attributable to scanners, since these are healthy brains, that are of a magnitude greater than that we observe in early stages of Alzheimer's disease. And this already sounds pretty problematic, but it's actually especially problematic in this particular context because these scanner effects actually overlap with the effects of interest. particularly, again, in kind of temporal lobe in both the medial and lateral sides. So this is a major issue. And this is kind of the main motivation for this project, which looked at how we can address this kind of problem. So to kind of contextualize our solution, we were looking at the Alzheimer's Disease Neuroimaging Initiative, which is still one of the largest kind of nationwide and including sites in Canada that look at Alzheimer's disease patients. And we particularly wanted to look at an early phase of the ADNI called the ADNI-1 because they collected T1 weighted scans at both 1.5 and 3 Tesla scanners, so differences in magnetic field strength, which would contribute even more to kind of some of these scanner differences. And this particular phase of the study involved 64 distinct scanners and included 3 different manufacturers. So we kind of expected to see, because of magnetic field strengths, as well as the sheer number of different scanners, that there'd be pretty notable differences across these study sites. So in order to look at this, we designed a kind of machine learning experiment because we wanted to see if we could detect which scanner, which manufacturer that the images were acquired on just based on their cortical thickness. And the paradigm is fairly straightforward. We just randomly split the subjects into 50% training, 50% validation, and we use a random forest classifier to try to detect if the subjects were acquired on a Siemens scanner. And we're just assessing this using AUC and repeating this 100 times over random sampling. And so what we found was quite striking in the ADNI-1 sample is that prior to performing any kind of harmonization method just in the original data, we were able to detect the Siemens scanner with an AUC of around 0.9 across the samples. And that indicates that it's very kind of easy in a sense to detect these scanner effects. And we applied a well-known and kind of in standard use method called combat to address some of these harmonization needs. And what we found is that combat performs quite well, reducing the AUC to about a little under 0.7, but that's still indicating that we can still detect Siemens even after applying the combat methodology. And so we looked at this gap and we suggested a new method that specifically leverages the covariance structure in these images. And we developed a method called CoVBAT, which was able to reduce the AUC close to 0.5, which is around chance. So indicating that we were able to kind of successfully remove these scanner effects. So I'm going to go into some detail, first introduce combat to introduce to those that are not familiar, and then I'll kind of build on combat to kind of explain the CoVBAT model. So combating batch effects or combat is a method that is pretty intuitive for removing batch effects. It came from the omics literature, and what it kind of seeks to do is use a relatively straightforward model in order to, first of all, estimate biological effects of interest with the goal of not removing those biological effects as we do the harmonization. And then the underlying assumption is that the side effect can impact the location and scale of the measurements of interest. And this model is assumed on every single region of the brain individually. So in this context, we're looking at cortical thickness. So we're saying that scanner can influence the mean and the variance of these cortical thicknesses at each region of the brain. And the goal of harmonization here is just to bring the kind of site-specific means and variances closer to the pooled mean and variance across sites. So that's our target. Just by looking across the whole population, we look at the mean and variance. That's kind of the target that combat has. Now, of course, you can define other targets depending on your study design. And the way that combat actually kind of estimates these parameters is quite straightforward. It uses a linear model and uses a location scale model in order to estimate the side effect parameters. So the assumptions in the combat model are, first of all, that the errors are normally distributed. This is using standard linear models. It's assuming the linear model with respect to covariates, and it's assuming that the side effects are kind of captured by location and scale shifts on each feature individually. While it is borrowing information across kind of the features, it's assuming that the side effect is specific to each feature, and it's just the location scale of each of those features. And so what we notice is that implicitly it's saying that there's actually no side effect in the covariances. Right? So if, for example, that scanner could impact the right-left hemisphere correlations, then combat would not be able to pick up on that kind of signal and would not address any side effect in that structure. And we actually wanted to look at this to see if this assumption was violated in this context. So again, back to the NE1 study, we noticed that prior to performing any harmonization, this is the three largest scanners in the NE1 study, we noticed that there's pretty striking differences in the correlation structure between scanner A versus scanner B and C, where scanner A is a Siemens scanner and scanner B and C are both General Electric. This kind of highlighted in general kind of higher positive correlations in a Siemens scanner versus the GE scanner. So it's a pretty striking difference that we thought may be contributing to some of these machine learning results that we were looking at. And what's also, and when we applied combat and looked at these correlation structures again, you'll notice that they don't change at all. So combat is kind of, again, is not addressing these specific type of side effects. And so we would design a method that specifically addresses this. So our method called correcting covariance batch effects, or COVBAT, is still assuming the same kind of combat location scale model because we want to preserve kind of linear biological effects, and as well as dealing with some of those location and scale effects that are well documented throughout the literature. But the main difference is that we no longer assume that the error vectors have the same covariance across sites. Now we're saying that these error vectors may have different covariance across site, and that's what we observed in the data, and that's kind of specifically what we want to address through our method. And so the goal here is analogous to combat, except here we want to shift the site-specific covariance structures towards the pooled covariance across sites. And the idea of how we did this is, first of all, looking at the pooled covariance structure and trying to identify structures. And so we do this using principal component analysis, which just breaks it up into kind of weighted sum of, rank 1 structures. And when we look through the rank 1 structures, we found that they're kind of consistent with the side effects that we're seeing. In particular, the first component seems to describe generally higher or lower positive correlations. And if we were able to address the first component, we may be able to address some of the side effects that we observed. And I also highlight this. There's kind of finer, still rank 1 components that we can address, and that there may be side effects in some of these structures as well. But just looking at this in particular, we thought that if we were able to shift these specific structures, we may be able to address the side effect and correlation. So what we did is actually we fixed these structures that are estimated from the pool covariance structure, and we estimate the site-specific covariances based on that fixed structure. So now you'll notice that the weights are now lambda sub ik. So there's site-specific weights on each of these structures. And when we do this, we are then able to shift those weights, the site-specific weights, towards the pooled weights. And a tractable way to do this is just to adjust the variance of the scores, of the PC scores at each site, towards the pooled score variance across sites. And what this would do is allow us to shift those site-specific weights towards that toward the pooled weights. And our new method here assumes still normally distributed errors with now with site-specific covariance. We're still maintaining the same assumption of the linear model with respect to covariance. And now we're making an additional assumption that our side effects are captured by population PC directions. So in the paper, we go through and evaluate cases where that last assumption may be violated and assess performance of those cases. For this presentation, I won't have enough time to go through all those details. So, but essentially just wanted to highlight that even when the assumption is violated, we still find that CodeBAS seems to perform quite well. So we applied CodeBAT to the EDNI1 data, and we saw that the kind of striking site effects in correlations were largely mitigated after applying CodeBAT adjustment, but still preserving some of the site-specific structure, which may be tied to biology. And when we came back to the sample, which consists of 505 subjects acquired across 64 scanners, just some demographic characteristics here, we wanted to revisit the kind of machine. We wanted to kind of come up with some more statistical tests to see if we were able to remove some of these side effects. So the first one we used kind of a traditional test, just a multivariate analysis of variance. And we found that prior to performing harmonization, that there's kind of a significant effect of manufacture across the sample. And once we apply combat, which doesn't really address the multivariate effects, that we still see that significant side effect. And finally, after applying our method, we're able to kind of achieve a non-significant test result for manufacturer while still maintaining significant associations with sex and diagnosis. And when we designed some more machine learning evaluations, we revisited the old one. Again, we finding that the AUC is considerably reduced in detecting of Siemens, so reducing the side effect. We designed other experiments to show that it maintains detection of Alzheimer's disease in this population, as well as maintaining some of the biological sex effects that are well documented in the sample. And so with that, we were able to actually show through our analysis that CoVAS seems to address the side effect issue that we observed in the AD1 study. And so I'm happy to take questions on that maybe towards the end. Let me just quick check on time. Yeah, okay. And so I think I'll have enough time to present kind of another kind of harmonization paper, but this time in the context of covariance valued measures in functional neuroimaging. And so happy to take questions later about the first topic, and I'm just going to go through some of the details here. Okay, so again, um, uh, coming back to a motivation here. So the idea of functional neuroimaging is that we're trying to get at how the brain is functionally organized. So it's well known through older neuroscientific literature that the brain is functionally organized into certain subnetworks. And these subnetworks have kind of specific roles and can be impacted differentially in certain disorders. So we can kind of conceptualize that in just the schematic diagram, but we can also estimate it using functional neuroimaging and kind of get a sense of the subnetworks that exist in this case, in a healthy brain. What I wanted to highlight is tie it back to Alzheimer's disease. This study not only looked at cognitively normal individuals, but also looked at Alzheimer's disease brain. And they found that these subnetworks seem to be quite disrupted in Alzheimer's disease, some of them no longer existing, and others kind of more functionally distinct based on these neuroimaging, kind of reflecting functional differences across the course of the disease, as well as reflecting some of the structural damage that occurs in these brain connections during Alzheimer's disease. So it's important both in studying kind of healthy individuals as well as studying disease to consider how the brain organization may be modified. And I just wanted to go through and highlight how these are typically estimated. So for those that don't do too much functional imaging or don't specifically look at this kind of outcome, so we start from these resting state functional MRI scans, which are acquired over time. Many scans acquired over time that kind of capture the brain activation. And then typically there's some kind of parcellations that's defined. So kind of looking at specific regions of the brain. And then at each region of the brain, we want to look at the kind of time series data from the functional imaging. And then what we're particularly interested in when it comes to constructing brain networks is to look at the relationship between these brain regions. And so what we do is we take this bold time series and we kind of derive a correlation matrix, which will tell us how these brain regions are activating, co-activating over time. These correlations are then transformed into connectivity values, which can end up being weights or just like either indicating the weight of edges in a network or the existence of edges in a network. And then we kind of identify brain networks based on that information, and we're able to do something called subnetwork mining or community detection in order to derive brain subnetworks. And that can tell us something about different populations and can also just tell us something about the typical functioning of a brain. So community detection has a lot of different methods for handling it. So for this presentation, I'm going to talk specifically about methods that identify brain subnetworks based on a quantity called modularity. And modularity is somewhat intuitive. The idea is that modularity is kind of like a goodness of fit. That if this specific subnetwork structure has high modularity, it means that there are a lot of connections within the subnetworks and not many connections between the subnetworks. So here in the red and yellow, you can see that's kind of high modularity. And the opposite, low modularity, is where the green and blue networks don't seem to have any particular pattern. They're not more connected within each other, and they're not less connected between. So we consider that low modularity. So there's a lot of different algorithms for this. We'll be using the Louvain algorithm to identify subnetworks that maximize modularity. And that's how we derive our brain subnetworks. But what hasn't been considered in the literature so far when it comes to brain subnetwork analysis is if sight can impact, so differences in scanner, as we noted before in structural neuroimaging, can those differences in scanner impact these studies of brain subnetworks? So we designed an experiment where we kind of looked, first of all, at aggregating different kind of functional connectivity matrices into like site average connectivities, and then deriving specific subnetwork structure for each site. And so here we have 4 sites, 2 different scanners across 4 different locations. And we found that the side effects seem to be pretty pronounced when it came to the subnetwork detection. That in particular, the difference between Cardio 1 here and other sites is quite striking, that we identify a very different subnetwork structure uh, that can be attributed to scanner differences. And what's particularly striking is actually the BSLA and Cardia are the same manufacturer and model of scanner, just at 2 different locations. So these are pretty striking differences when we look at a population level, which is a kind of which is quite often how we look at these in these kind of studies. But I also wanted to look at a little more detail to see if we can detect these differences at an individual level. So if for each subject we're able to derive a brain subnetwork that is kind of personalized and individual for that individual. And what I did is kind of adapt a hypothesis testing procedure that uses information contained in pairs of these networks. So if we're able to define a distance measure to kind of describe how every individual is different from one another, we can actually use that distance information to do inference. So here we use a measure called the variation of information, which is a valid distance measure on these kind of space of possible subnetworks. And what we do is we look at these distances and we want to ask the question, are these subject-specific subnetworks different across the population? And what we found is that is yes, that there is a striking difference. And so here, this is kind of a plot where we transform the p-values such that higher and anything above the blue line is indicating a significant effect. And so here we see in our sample that there's a significant effect of scanner, as well as agent sex in the kind of individual subnetworks collected for each individual. And so from here, we really wanted to address this problem because these kind of side effects could impact the downstream analyses when we're looking at subnetworks, both at a population and an individual level. So what we did is we actually adapted. We in the previously they had adapted combat for this kind of analysis, where they applied combat to the connectivity matrices and found that it kind of reduced side effects in several network metrics. However, in this particular context, we hypothesized that this FC combat adaptation may not fully address our problem because what we're really looking at is kind of correlations among edges, right? So like which kind of groups of edges are connected with each other, and the kind of correlation among those groups of edges. So we thought that it may be important to consider that kind of correlation. So in order to do this, we proposed 2 extensions, one of which kind of used borrowed information from the atlas that we defined in the parcellation as prior subnetwork information, which can inform the kind of blocks that we're interested in considering. And the other main method that we used is kind of extending CoVBAT to the setting, where we use CoVBAT to harmonize the covariance structure across the edge weights. And then we wanted to see if this could address some of the issues that we saw. And so we applied these methods and compared them in a sample that consists of 2 different studies. One is the BSLA, the Baltimore Longitudinal Study of Aging, and the other is Cardia, which involves a younger adult population. And so you'll see here that there are 2 different manufacturers / makes of scanners at 4 different locations. And in particular, you'll also notice that since these studies have different designs, that there's a kind of difference in the age range that were acquired across these samples. But we're mainly looking at this in the context of 4 different scanners, and we're evaluating our methods in these 2 large resting state fMRI studies. So when we applied our methods, we found that the striking difference in the population level subnetworks was largely mitigated using any of the 3 methods considered. So we found that FC combat, block combat, FC combat, they all performed quite well for reducing this specific difference that we saw. The subnetwork seemed to be more harmonized after applying. And then when we return to the individual level analysis, again using a variation of information, and here using a test called multivariate distance matrix regression, which allows us to do a regression by leveraging this distance information. And so this is actually a kind of test that we designed because previously in the literature there had been very few cases where people looked at kind of individual level subnetworks and trying to identify and test for specific effects of interest in that context. So from this test we designed, we applied our three harmonization methods, and we found that, again, initially that there was kind of a striking significant difference across sites, across scanners. And we also found that after applying combat in this context, we weren't able to reduce this specific type of side effect. And this was also true of our block combat method which we proposed. But we're very pleased to see that the FC combat method actually reduces this specific type of side effect to the point where it's no longer significant through MDMR testing. And then finally, we examined several network metrics that are related to subnetwork structure, things like modularity when it comes to the atlas, modularity for the kind of the modularity detected across the sample, and also the clustering coefficient. And we found that across these network metrics that we consider, that all the harmonization methods seem to perform quite well, and that all of them largely remove the side effects that are significant in the unharmonized data, while preserving the age effects that we were able to note in the population. And so with that, I guess it's probably a good time to kind of wrap up. So those are kind of two main projects, and I have another project where we looked at inference on multiple data types, and I'm kind of very interested in going towards that direction. So if anyone's working on kind of multimodal data analysis in this context, or really any context, please feel free to reach out. Certainly, any interest in harmonization, again, feel free to reach out. And thank you for your attention. And I'm happy to take any questions and participate in the working group discussion. So thank you.